On that note, I wanted to move to the next speaker. Even though this meeting is really focused on biofoundries and their applications and et cetera, et cetera, et cetera, I wanted to make sure that we had a bit of a snapshot as to what SynBio is in Canada today and where it's going to. So I've asked Michael Dorrington from the Ontario Genomics, or I'm not going to go into the details of how genome works in Canada, but in Ontario, the province next to ours, there's also a very, very big push around synthetic biology. And Michael's group over there has been very central to pushing the SynBio field in Canada. So they've been hosting a lot of meetings and putting out a lot of white papers on visions and things that need to be done in Canada to move SynBio forward. So I've asked Michael to address the audience for a little while to tell us about what's going on in SynBio in Canada. Thanks, Michael.
Thank you. There we go. Perfect. So I was reminded last night that we really need to start developing some self-generating textiles because I burned a hole in my shirt with the hotel iron, which was a great start to my weekend here, but hopefully it's only on the way up from there. Okay, so good morning, everybody. As Vince mentioned, my name is Mike Dorrington. I'm an advisor at Ontario Genomics, and it's really an honor to represent OG this morning and talk to such a distinguished crowd. I'm just going to talk for 5 minutes about our current and future work in building a national synthetic biology ecosystem here in Canada. You know, we at OG understand, as I'm sure all of you do, how important these platform technologies are to addressing some of the major issues of our time, and we're really all about bringing this community together here in Canada. So our vision for this is really based on two key phenomena that will be very that won't be any surprise to any of you. The first, of course, is the dual crises of our climate and waste, and so it's become obvious that we really need to shift from our current create, use, dispose economy to a more circular economy, wherein we can take waste from various processes and turn them into value-added products. And synthetic biology is absolutely fundamental to being able to do this. In order to do this, though, we're going to need to really build more connection among various groups of people, and that includes academic researchers, industry professionals, national and international regulators, as well as consumers themselves, and having everybody kind of understand how this chain works and how it can contribute to a greater society. Now, as was just mentioned, this is also going to require significantly increased coordination and communication between economic sectors. So synthetic biology can be that platform technology that really links things like health, manufacturing, agriculture and food, and other sectors, and share information to create a more flexible, resilient, and efficient world economy. Now, Canada has some tremendous advantages to making this happen. So we have world-class academic institutions, including Concordia, and represented here by my alma mater, McMaster University. We have a strong history of manufacturing innovation. We have strong life science research expertise, and we have a nearly unmatched access to carbon sources through our natural resources and agriculture industries. So all of these things can converge to make Canada a potential powerhouse in the emerging bioeconomy. Now, bringing all this together requires strong leadership, and that's why OG has gone and created the Canadian Council for Engineering Biology. This is a collection of experts from academia, private industry, government, and even finance, and includes Vince himself. And these have all been handpicked to leverage Canada's advantages and bring about this Canadian biorevolution. So our first steps have been geared towards community building. I don't know why. There it is. Community building and thought leadership. So beginning in 2018, we've hosted the annual Canada SynBio Conference in downtown Toronto. This has brought together the national SynBio community to share knowledge and highlight opportunities and obstacles within this community. The COVID-19 pandemic, of course, necessitated a shift to more online programming, which led to our 2020 webinar series. And all of these webinars are available on demand on our website if anybody's interested. And then last year, we were delighted to welcome people back to Toronto for Canada SynBio 2022. This was a stellar event headlined by Paul Fremont himself that highlighted how far we have come despite and in some ways aided by the pandemic, right? It's really brought some of these issues to the forefront of people's minds. Building on this, we've also published 2 groundbreaking reports. So first, in 2020, the Council published our white paper on the potential power of synthetic biology and how it can revolutionize the Canadian economy while benefiting our environment and society at large. This was followed up last year when we published an in-depth report on cellular agriculture, something that we're very interested in. And included a great in-depth economic analysis by the University of Guelph's Mike von Massow. And together, these reports really represent both a look backward at how far we've come up to this point, but also more importantly, a look forward at what is possible. So more than anything, these exercises and our interactions with the Canadian SynBio community have shown that there's a lot of work to do. So this needs to start with some strong road mapping and continuous conversation with governments at all levels. And we know that some of our messaging is breaking through with our national government, at least. New funding streams are being announced. The dual Canadian national biomanufacturing and genomics strategies, as well as many conversations with government around access to infrastructure and talent development happening as we speak. So through groups like the Canadian Council of Engineering Biology, the Global Biofoundries Alliance, and others, we can really take this biorevolution to the next level and make it successful here in Canada. And we're so excited to see that. Finally, I'd be remiss if I didn't give a quick plug to our 5-year anniversary of Canada SynBio. So again, in Toronto, we'll be hosting any who want to join us. It'll be in early June of 2023. We'd love to see many of you there. And I know this has been a very quick rundown of what we're up to, so I encourage anybody in the audience today to come find me if you want to delve a little deeper into what's happening in Canada and what our future goals are. So I'm really looking forward to the rest of the meeting here and really excited to learn a lot and see what's happening around the globe. So thanks, Vince. Thank you, everybody. And let's have a great meeting.
Yeah, so hi everyone, my name is Evelyn Trafnik. I'm the principal investigator for Lifelike, which is funded by the Novo Nordisk Foundation and the Center for Biosustainability located at the Technical University of Denmark. And I have a team at UCSD inside the Systems Biology Research Group in bioengineering and also a small team in Denmark. I also used to be the Chief Information Officer at the Center for Biosustainability for about 4.5 years. I was then recruited into Join Bio, leading digital science and technology team working on nitrogen fixation. So today I would like to show you a product called Lifelike. It is an open source platform and it's about knowledge mining and data storytelling. So first, a quick introduction on my product portfolio. I joined a company called Genomatica, which I'm sure most of you heard of. They produced one of the first commodity chemicals in E. coli, bio-based BDO. It was spun out of UCSD with Dr. Bernard Bolson and Christoph Schilling as the founders, and I was one of the early employees. And there we actually started out working on a commercial metabolic modeling and simulation software. This was at that point very ahead of its time, and it was right when Dr. Bernard Bolson invented kind of like flux balance analysis. And so we first thought we could commercialize that, but it was really early. So the company decided to take it off the market and actually apply it as a competitive advantage to the development of biochemicals. At that point, there was a need then to figure out how can we produce chemicals in an organism. So we built another platform for the identification of biotransformation routes, and which allowed us to establish a comprehensive patent portfolio. And then as we added a lab, we also needed the LIMS. And back then you couldn't really buy those. So we built a very comprehensive LIMS supporting the entire DBT cycle. Then I joined the Novo Nordisk Foundation Center for Biosustainability as the CIO. And there, besides supporting and implementing all of these same type of solutions, I also started thinking about a problem that I did not quite have a chance to address at Genomatica. And that was, of course, not unique to Genomatica. It's a problem we all of us have. So we were doing a lot of like omics data analysis. We were applying systems biology, and the systems team would analyze the data and create slide decks and create reports. And it occurred to me that all this valuable information got lost when people left the team. The knowledge would walk out the door when they did, right? It was hard to find, hard to reverse engineer, yet it was all this great knowledge that gives a scientist experience. So I wanted to address the reuse of knowledge, giving the machines also the opportunity to remember knowledge. And that's when the idea of Lifelike was born, and started conceptualizing it, getting funding for it. So what exactly is Lifelike and how does it work? We feed Lifelike with knowledge from many public databases, which you can see here in the picture. And once Lifelike has all this knowledge, it can assist us with our research. It can help us to read, understand, and enrich unstructured text and structured data. So users can upload any PDF, and like Lifelike will detect known entities from the text, classify them into different categories such as genes, proteins, diseases, chemicals, etc., and will link them to the public databases where further information can be found. This is called entity recognition. We are now able to consume this PDF more efficiently. We can use entity clouds to see the big picture that's being discussed, and we can navigate through that paper. We can extract also knowledge from it, and as we do that, we're creating data stories. And so it has these data storytelling capabilities. For example, you can drag knowledge onto a canvas, and I will show you in a moment. We can also give Lifelike structured data, like an experimental data set, such as an RNA-seq data set, proteomics, metabolomics, and Lifelike will then generate basically a story for us by contextualizing the data against a chosen knowledge base, for example, a knowledge base like Biopsych or Reactome. And it will actually tell you what are the most relevant things that you should consider. It's kind of like a graph-based search engine. So why did we build Lifelike and knowledge mining? What are the problems and opportunities? As I've already mentioned, one of the most common forms of knowledge sharing is through reports, slide decks, or publishing, right? And the sheer volume of that is overwhelming, and you know, we wish we could speed read, right? And while a publication is considered knowledge for humans because we can read it, make cross connection, take away just the right amount and draw conclusions, for a computer, it is just information, right? It is because every word has the same weight, and reverse engineering text are our hardest AI problems. So another form of sharing knowledge is public databases. And there we have a huge amount of information silos. And here we have the opportunity to magnify their value if we connect them. We're basically drowning in information, but starved for knowledge. This is a great way to put it, and that is the problem that we're trying to solve here. So there is yet a third problem that needs addressing, and that is complexity. We have some really great curated pathway databases like Biopsych and Reactome. We're finding that when we mine them, the complexity explodes very quickly. We get hairballs, we can't really understand anything anymore. And so all of these problems make it very hard for us to be able to see the big picture and find the most relevant information. At the same time, I believe that we have a great opportunity to accelerate research if we can solve this. And we have done that in Lifelike with text mining to address the first problem, building an integrated knowledge graph. And with graph algorithms and special visualization techniques, we can show you the most relevant information and allow you to zoom in and out of that knowledge. So knowledge graphs is the name of the technology that we're using here. And how does this fit in with other techniques? On the right-hand side, we have full mechanistic models that are very much white box analysis techniques. We understand how something works. We understand why it works. But the problem is that we cannot reconstruct these models for the entire cell. We can only do it for metabolism or a pathway, so it's limited in scope. Then as we generated more and more data, we started jumping into machine learning. This is a very much black box analysis technique. We get a result back, but we don't understand why and what it is. And so knowledge graphs actually bridges the gap between the two. They can help us understand the AI results, providing explanatory AI. We can also actually extract new features for AI out of knowledge graphs. So knowledge graphs are basically changing AI by providing context. So how do we do this? So we bring together these various databases and we put them into an integrated knowledge graph and store it in a graph database. And so this graph now contains many different things like NCBI genes, KEBI, biopsych, et cetera, et cetera. We also have a literature extraction data set from where we can, where we store relationships between genes, chemicals, diseases, and so on. So now that we have this, what I like to call digital mind, I will show you how we can perform a semantic analysis on PDFs. That's like the first thing that we can do. How we can extract knowledge from many different databases using like an enrichment table, how we can do statistical enrichment against ontologies, how we can do graph analytics against it. And then finally, as we learn new things, how we can actually create knowledge graphs. So let's have a look. You can upload any PDF and Lifelike will use AI and rule-based entity recognition to identify. So let me show you here. So here's a paper, here's an example. It will show you we are identifying anatomy terms, chemicals, diseases, genes, phenotypes, proteins, species, etc. And so you can click on these terms that it highlights, and you can then link to a database where you can find more information about it. So you can kind of go through the document. You can, we're giving these terms an identity. We also have this entity cloud. And so, for example, if we want to know where in the document is a term being discussed, we can go through it and step through it and we'll recognize synonyms. And so this is kind of like a way we can now more easily understand this document and navigate through this document. Okay, so now I will show you how you can also extract knowledge from this document and perform searches against other information in the knowledge base. So let's have a look how this works. So here I have again a paper. I'm grabbing that paper, putting it on a canvas. I want to remember it. I'm also grabbing entities that were recognized in the paper. I'm simply just dragging them over. I can highlight text and say, oh, I'd like the snippet of text. Let me bring it over onto my canvas. I want to remember where this came from. Every term that you drag, it will remember where it came from. So at any time when you click then the link again, it will take you back to that. So now I'll grab a couple of genes out of the document. And so now maybe I want to know what is the literature discussing about this gene? What are maybe the associated diseases? So I will click on, say, knowledge graph search. It immediately executes a search. I find the gene. I see, yes, there is literature information. And then I'm going to say, I want to see all associated diseases. So now it extracts all the diseases that are being discussed in relationship to this particular gene. This could be phenotypes, this could be other things, but so then I can see how many snippets are supporting this relationship. I can look at those snippets and I can click on a paper and look at the snippet itself to see how is it supported, so I can quickly scan through it. And let's say I like to capture this information. I again bring it over, I associate it. And if I want to remember and provide evidence, I can just grab that snippet and paper and provide it as evidence on that relationship. So now I might want to see, oops, I'm so sorry. So now I might want to see how, if anybody has built any other maps. And so I can click another button and see, are there any other maps that have been built that contain this gene? And if I like it, I can again just drag it. So everything is like a drag, drop in Lifelike. And then I might want to do another quick search and say, are there any other documents that discuss this gene? So I can see the documents that are already in Lifelike. I can click on them, get a bit of a preview. And if I like the document, again, I can grab it and put it on the canvas. So, and then if you, like I already said, everything is hyperlinked. So basically, if I want to know where did this snippet come from, this text come from, you click on it, it opens up that document. Okay. So before I show you the next features, which is the creation of enrichment tables and graph analysis, I would like to introduce you to a use case. So here we were analyzing a strain crash. The strain was growing, producing melatonin, and at some point it crashed. And so we used RNA-seq data to debug this. We took 2 time points in the crash zone and calculated the fold change and then analyzed that data with enrichment table and graph analysis. And let me show you now how that works. So one great feature of this integrated knowledge base is that you can retrieve information from many different sources on a button click. And in addition, we can perform statistical enrichment against different ontologies. So here is the example for the strain crash data. You simply copy the genes into a table and then Lifelike will pull information from sources like NCBI, Regulon, if it's E. coli, UniProt, string database, etc. And then we can do, again, an entity recognition on the text. So we're again identifying known entities in the text and color coding them. And so here you can see that when you then click a button, see again the entity cloud that zinc stands out and this regulator here, right? So you can again see that based on the semantic analysis, there's zinc apparently something that is relevant. Then we have the ability on a button click to perform an enrichment analysis against the Go ontology. And so we can see here that also cellular response to zinc ion starvation is statistically enriched and the associated genes. And so we're getting the same answer, although it's coming from different knowledge sources. This is coming from Go ontology, and this is coming from textual descriptions out of different databases. So the same here with the downregulated genes in this particular data set. And over here, we can see putrescine catabolic process, again, statistically enriched, as well as standing out in the semantic analysis. And so we're going to remember now this putrescine, because in a moment, I'm going to show you how we can then see more information with graph analysis. So now, now we're going to have a look at knowledge mining using graph algorithms. Here, I'm going to show you how you can contextualize experimental data against pathway databases like Biopsych or Reactome. Biopsych is mostly microbial, and Reactome contains mostly human biology. These databases contain metabolic pathways, as well as biochemical and transcriptional regulation, and in the case of Reactome, also signal transduction. So when we do graph analytics, we're basically traversing these networks from different starting points based on a particular question we want to answer or based on a particular expression data set. So let me show you conceptually how this works. So here we have a graph that contains, let's say, biochemical reactions and regulatory relationships. And let's say we have now starting points in this space where this could be RNA-seq data. So what we then do, we call this analysis a radiate analysis, because we're essentially like analyzing the neighborhood around those. And we're going in a backwards direction and ranking every node in relationship to these starting points and in terms of how relevant it is. And we're doing the same in a forward direction. And then we end up with a ranking sheet in a forward and backwards direction. And we can say, what is influencing our genes and what are our genes influencing? And then we can pick certain endpoints and we can say, then visualize it. And here we had to come up with our own visualization techniques. There was nothing out there that we could use. Cytoscape was not able to help us. And the company, also worked with like a graph database vendor. They were not able to help us. So we came up with this unique Sankey-like visualization so we could cut through the hairballs. For our particular use case, now let's narrow in again on zinc. So zinc stood out here again as a node. Relevant, you should pay attention to this. So now we can say, well, how does zinc connect to our starting points? And so here we can see every gene in this data set has a color, and we can see how all of it connects to zinc. And some of the zinc-related genes here we're seeing in a close proximity, and they are highly upregulated. The height of the bar shows us like the level of expression value. And we can, see other relationships, not just the zinc relationship here, but also like the relationship to iron, spermidine, arginine, other things here. You get the picture that you can, see the connectivity, right, and understand the mechanisms. The same here in the reverse direction. So here we're asking how is zinc affecting the genes, and so we can in a reverse direction see that it's highly connected to these highly up regulated genes. And in this visualization technique, it's color coded, but in another visualization technique, we can actually see which relationships are regulatory. And so we can see here that this regulator inhibits this gene, and then this is then under zinc deficient conditions actually highly upregulated. So, and then let's remember from earlier, we saw putrescine. And so if we ask the question, how is putrescine related to all of these genes, we actually nicely then see the relationship to zinc from putrescine, but we can also now see that it activates SOX-R and it then activates SOX-S, which then activates ZIN-T. And so we can derive from that maybe putrescine might contribute to the high activation of ZIN-T, in this case, through SOX-R and SOX-S. So here another example for this use case, this is from the human space. We had metabolite measurements from sepsis patients. So there was a group of patients that had high mortality and a group of patients that had low mortality or no mortality. And so we looked at the metabolites that were significantly different between the two groups and analyzed them against the Reactome database. And from that, we then asked the question if it plays a role in sepsis. So we found in this case a particular protein, this particular protein here. And then, so it informed us about this protein being relevant. And then when you ask the question, does it play a role in sepsis? It very much does. It actually plays a very crucial role. The other thing we then did, we had a data set of about 50 papers where we extracted relationships between entities out of there. And we asked that data set the question from common plasma metabolites, what are the shortest paths to mortality? And so we generated that result and so put it into Lifelike. And at this point, when we had this protein and investigated it, we came across again that analysis, and we were able to see it in between carnitine and mortality. So this particular protein was in the middle there. And so now we're being informed that is carnitine potentially related to mortality, right? And so basically, we can infer knowledge from this, and it very much is. And the next thing, when you then ask how it's related to sepsis, we found that there's already a clinical trial on that. So Lifelike informs you about the relevant information. It takes you very quickly to the most relevant information. So basically, the other thing we're seeing here, we only had 9 plasma metabolites, and we were able to gain so much insight, right? So we basically are moving from big data to getting big insights from small data. So now, real quick, Lifelike is very rich in data storytelling features. You can build large knowledge maps, and then you can also export them as PDF and send them to collaborators. So here's, for example, a map, and just shows you how you can link a lot of information together. So you can link to enrichment tables. You can simply drag them onto the canvas. You can have pictures. You can snip pictures. You can link to papers, other maps, network traces, etc. So another example that is super useful is actually patent analysis. You can bring a patent in here as a PDF and then analyze it. And we all know how difficult it is to understand patents. So you can actually, drag again entities onto the canvas and reconstruct that patent and try to understand it. So, so what is our vision here? We want to make a public deployment with self-registration. So currently you can partner with us, but we also want to support scientists around the world who are solving big world problems and help them to maximize the reuse of billions of research dollars, find new unintuitive connections, to see the big picture and the full picture, and basically gain big insights from even small data sets. So how can we accelerate this? We will, on our end, continue to demonstrate the value of knowledge mining with many more use cases. I would like to ask all of you to encourage you to write, please, machine readable papers, because you might be surprised, when you open them up in Lifelike, that it's maybe not machine readable as it could be. I also like to encourage everybody to fund and contribute to knowledge curation efforts, because they're so important. We can really utilize them here for knowledge mining. For example, you can actually adopt the biopsych strain database and contribute to the knowledge creation. So basically, instead of just publishing text, I would like to encourage people to think about also publish knowledge, and by contributing to knowledge creation efforts. So basically pay it forward, because Lifelike will pay you back when you're debugging the next train crash or wondering why one patient group is dying and another one surviving. If we can enable our computers to effectively reuse prior knowledge, then we can reach a new level of digital wisdom and build Lifelike virtual assistants that can accelerate research. So now open to questions.
Yeah, can you hear me, Evelyn?
Yes.
Yeah. I think that in the interest of time, we will keep the questions for the workshop afterwards. We are already running a little bit late. So, yeah, so I guess I'm next. Thank you. Can you switch? What? Okay. So today I would like to invite you to think about the way we are distributing the plasmids or the DNAs that we engineer. And the biofoundries, I really believe, are the incubators where the technologies that will catalyze the next industrial revolution are being developed. And this is really because we are changing the way we are thinking of the way we develop product in biotechnology. We are really moving away from the fishing expeditions that have given us the drugs that we are using today. And we understand that Apple doesn't release the iPhone every September by screening random assortment of components. So we're really trying really hard to learn how to adopt a model-driven product development cycle. And when we look at the details of that cycle, you have a combination of computational and physical workflows. So it's really a cyber-physical process. It's really known that those processes, maintaining their integrity is very difficult because you have a lot of point of vulnerabilities. And good cybersecurity, physical security hygiene goes a long way toward making those processes more resilient. But in those processes, there is something very special, which is DNA, because DNA has a dual nature. It exists as DNA sequences in computers, and it exists in the form of molecules in the physical world. And so we can go from DNA sequence in a computer to a physical sample through synthesis, and we can go the other way around through sequencing. And so a fundamental question that I think a lot of us are struggling with in one way or another is that we have DNA in our freezers, we have DNA in our computers. And if the samples that we are working with do not match what we think that they are, then all sorts of bad things can happen. The most likely outcome that our experiments don't work. But there are all sorts of other scenarios that we can think of that are a little bit more troubling or damaging. So why is it so difficult? Well, it's difficult because, remember the last time you bought a plasmid from Agene or from Fisher or whoever, you really pay money to get access to a physical tube that contains a DNA molecule. And in order to use the content of that tube, you need some documentation, and that documentation is generally freely available. Both the DNA and the documentation can be copied essentially at no cost to you. So there is a lot of plasmid sharing going on in the community. But the problem is that both the plasmid and the DNA can be edited. And so before you know it, you end up with people who have like a plasmid and the documentation that don't match anymore. And this is a problem. This is a problem for everybody. This is a problem for the developer of the plasmids because essentially they don't know who's using what they've done. There is potentially a loss of revenue if you're selling the plasmids. There is a loss of credit if you just want the credit, and you cannot provide tech support to your, so you cannot support your users. But it's also a problem for the plasmid users because even though they may get the plasmid very cheaply initially or for free, they're going to be wasting a lot of time and resources in experiments that don't work, in detective work to try to piece together the documentation of what they're working with. And if you're working in industry, you may be spending an awful lot of time figuring out if you have freedom to operate. So this is not a new problem. More mature industries have solved it by using by putting in place product identification systems. And the automotive industry is relying on the VIN system to essentially build a robust link between the manufacturer, the car owners, and a series of electronic records. And VINs are used every day. It would be very difficult to imagine the automotive industry without the VIN system. It's used to, issue title, collect licenses, insure your cars, etc. So the VIN number, vehicle identification number, is actually not a number, it's a certificate. It's a very simple digital certificate that includes multiple data blocks. And in the middle of that, the digit 9 in the middle is a security feature. So it's used to make it a little bit difficult to forge VIN numbers that don't exist, or it can also be used to catch transcription errors. So in our situation, we have the same need. We have developers of DNA molecules, we have users of DNA molecules, and we have a bunch of documents that are attached to those DNA molecules. And it's really, if the three work together, that it would be good to be able to relate all of them one together. But we don't have a system like this. And the reason why is that because we don't really have a way to identify the products that we make. So it is very common for people to say, well, what about the sequence, or what about the plasmid name? So I'm going to show you that these are not good identification systems. Like using the plasmid name as an identification, it's a little bit like saying, okay, look, I'm going to put a Ferrari logo on my Corolla. That doesn't make it a, that doesn't make it a Ferrari, right? So that's extremely naive to think that, if you have a tube and a file that have the same name, then they describe the same thing. The DNA sequence, that's a little bit like, okay, we are going to identify our cars using the blueprints. There are a lot of things that are very closely similar, and it doesn't solve the problem that, all the make and models have the same blueprints, yet you have individual products assigned to different people. So there is a serial number aspect that is missing. So we have developed a few years ago a software that generates digital certificate that we call a DNA identification number, for lack of a better word. And essentially what that software does is that, you upload the plasmid sequence on the server, and the server generates a new plasmid sequence with a fragment that is carrying some data that is essentially a certificate that is computer generated. So if we look at it in more details, that sequence is not a barcode, okay? It's much more sophisticated than a barcode. A barcode is essentially a unique random sequence that you can use to identify, but here there is a little bit more than that. First, there are multiple data blocks. We encode in the DNA sequence the identity of the author of the plasmid serial number, and we have two security features. One is a signature that makes it possible to really build a connection between the entire DNA sequence of the plasmid and the signature. So you cannot take that DIN number, put it in a different plasmid, and you can detect that. The other thing is that there is an error correction code. So, like transmission of DNA through generations, there is mutations. Sequencing is not, bulletproof, so you can have sequencing errors. And so it's important to have a security solution that is a little bit more tolerant of errors than, in a computer science application. So the error correction code makes it possible to say, okay, this is the right signature, this is this plasmid, but by the way, you have like 3 or 4 mutations that have appeared since it was signed. So a few years ago, we published a proof of concept work where we have actually inserted those certificates in a few bacterial expression vectors. We did different variants. We had the original plasmid, the plasmid with a certificate, and then we started introducing some mutations on purpose, both in the signature, in the coding sequence, and in different places. And we just wanted to see, like the obvious questions is, do a sanity check. Can we propagate the plasmid? Is there going to be, some issues with stability of the plasmid? Are we going to introduce, can we introduce those sequences without interfering with the biological function? And most importantly, can we sequence them well enough that we can actually recover and validate the certificate? And the answer is that, it passed the sanity check. We could propagate the plasmids. There was no obvious effect on the plasmid function. And in most cases, we could recover the certificate and validate the certificate using Illumina data. So it passed the sanity check, and it's time to go to the next step. There's been a number of publications already at different levels on this topic. And at this point, we are thinking about, well, how do we deploy that? We can think of, there are different deployments possible. I'm going to propose a couple. One is publishing of plasmids. So really, the way it would work is you upload the gene bank file on the server that is your plasmid and your documentation. The server would then, send you back a sequence that includes a DNA fragment that codes for a DOI. We'll keep the documentation on the server. You make your plasmid, you distribute your plasmid, you lose control of your plasmid because the plasmid is shared, through students, labs, and things like this. But at the end of the process, your user even though you may have lost track of the plasmid, your user can sequence it and upload just the raw sequencing data on the server, and the server would say, oh, this plasmid was developed by Joe, and by the way, here's the documentation that was submitted by Joe. So that's very much like a publishing model adapted to plasmids and their documentations, which would be a way for getting citations, getting credit, and things like this. The other solution, the other business model that we are looking at is actually selling plasmids. So in this case, you would have a developer who is like an author who would go to a company that would be publishing the sequence, would provide the documentation and potentially a DNA molecule. The publisher would issue the certificates, and then the end user would buy the plasmids from the publisher. The plasmid would add the certificate. The documentation would be copy protected. That's easy to prevent the copy of the documentation. The plasmid may circulate through the community, but then people would sequence it, upload the trace files, and be charged a fee for getting access to the documentation of their particular sample. So that's another approach. There are benefits both to the developers and to the authors. For the the engineers who develop the plasmids, you get notified when your plasmids are used. Essentially, every time somebody is uploading sequencing file, you can get citations, you can get reviews, all sorts of status symbols that you may be interested in, or you may get royalties, like like when you're publishing a textbook. The other thing is that you could control the information, the material. Like, for instance, you can send the same plasmid to Joe and Bob, but have different serial number and say, look, if you're leaking that one, you're going to owe me a royalty because I know it came from you, right? So that now you can have something like this. You can also control the release of information. Right now, we are living in a world where we're giving all the information with our plasmids. But you may just want to give a user manual without any sequence annotations, because the sequence annotations are kind of your trade secret. Okay, so that's in environments where you're working with things that are dangerous, that could also be a way of ensuring deniability. So if you certify everything that you're working with, and if the FBI is knocking at your door with another sample, you say, no, look, it's not mine because all of mine are serialized and I know what I'm working with. To the user, it's a clear identification. Now you're getting a plasmid, you know where you're getting it from, so you can trust that person. That's where you're going to go for having tech support. You can get instant access to the sequence and the documentation, so you're saving all that work, detective work, where you're essentially not trusting anything that you have. That's also something that you could be using, within an organization at different stages of your workflow. You could quality control your plasmids and making sure it's still the same. You could potentially get post-acquisition, post-purchase support. Like, if there is an updated data sheet, then, the author would be able to push that back to you. And if you're working in an environment where, IP matters, and you can have clear access to the IP status of that DNA sequence. And so my point is that, the DNA, as we think of it, the value is shifting. We are coming from a world where the physical sequence was really the bottleneck. So people were getting access to a DNA solution bacterial culture, and there was no copy protection. There is also no IP status for most of that, because some of them can be patented, but DNA sequences cannot be copyrighted by statutes. So essentially, if you cannot be patented, you're screwed, even though you're putting a lot of effort into it. On the other hand, the documentation is becoming more and more important as our constructs become, more sophisticated. And that's annotated sequences, user manual, data sheets, IP status. The documentation can be protected. It can be copyrighted, and it can be copy protected. So you can control the dissemination of your protection. And so I think that, when we're thinking about the way we are disseminating our plasmids today, I think we should be putting way more emphasis on, while putting together quality documentation where really the value is, and be thinking of ways of, building a hard link between the documentation and the DNA molecules that are in circulation in the So this is some of the people who have been involved in the bioinformatics experimental work, and my colleague in computer science who have done all the cryptographic work. And we got funding from NSF and IH and CSU VP of Research. And same thing, we'll be happy to take any question during the workshop afterwards. Yeah, come over. So Tim McRobin, your research fellow in Marcelo Group at the University of Queensland. And he's going to talk about model-guided pathway design.
Yeah, thank you. Let's see how this works. Okay, good morning. Yep, so my name's Tim, and I'm going to be talking about how we can use a bit of systems biology and leveraging multi-omics data in a biofoundry context. So just a brief overview, I want to talk about how biofoundries have really shifted the bottleneck in the design, build, test, learn cycle, and how learning still remains a key issue, and how systems biology and metabolic modeling are key tools that can be used to help facilitate learning and accelerate strain engineering, potentially. And I want to illustrate some of this through an Amorous collaboration case study. So there's no question that biofoundries have really revolutionized how we do metabolic engineering. They've really spawned the design, build, test, learn mentality. They've obviously debottlenecked the build part of the cycle, but as I said, learning still remains a key issue. So they haven't necessarily accelerated the full cycle of strain engineering. We also have troubles with efficient data integration. So just to illustrate this, I just want to speak about the design space challenge. So as synthetic biologists, we're continually trying to chase longer, more complicated biosynthetic targets. So back in 2015, people were making opioids in yeast that consisted of 20 steps out from central carbon metabolism of native and non-native yeast genes. Just recently, Jay Kiesling has published the anti-cancer vinblastine in yeast that consisted of 30 non-native enzymatic steps extending from not so central carbon metabolism, out of the mevalonate pathway and tryptophan out of the shikimate pathway. So if you consider a foundry which may be able to say make 1,200 strains rationally a month, and if you look at a pathway with 20 genes, and you might consider expressing those genes at 5 different expression levels, whether it's by different promoters or copy numbers, that would lead to a combinatorial space that would take 6.5 billion years to synthesize. Now, if you consider that you might have a library of 2,000 candidates for an individual gene, that number just goes astronomical. So you start to need approaches to really navigate this design space. So people have been doing it in different ways. There's the more naive statistical design of experiment approach. The difficult with this is it's a very complicated relationship between gene expression and your end product titer, for example. And that tends to lead to poor performance of the methodology. Another approach is random mutagenesis or ALE. And if ALE works, it works, and there's nothing that can really beat that. But there's very few instances where you can actually apply that. In terms of random mutagenesis, the sort of workflow might be you mutate your strains, you screen them in a high throughput manner, probably in a plate format. You do endpoint analysis, and then you sequence your strains. You try and use machine learning to correlate what SNPs have led to the improvement in the phenotype. But there's some problems with that approach too. Learning still remains difficult. SNPs are information sparse. So, phenotypic changes will only be observable with the correct number of SNPs in the correct order. You need to debottleneck the pathway, right? And this can lead to limited usefulness of the prior data. So one potential approach to this is using systems biology. And this is becoming increasingly more feasible with automation and high throughput, low cost omics data. So this is just how a systems biology workflow would compare to, say, the random mutagenesis type approach. So instead of using plate format, you'd probably need to increase the volume of your reactor system. You do a full phenotypic characterization of the cells, and then you would have to integrate the data use modeling to then predict new strain designs, and the cycle will continue. So I just want to talk about some of the challenges we face transforming ourselves for the Amherst collaboration. So we needed the capacity to be able to do the reproducible characterization of a large number of strains, to be able to prepare and process a large number of omics samples, to handle all this data and to somehow learn from it. So to do this, we decided we really needed to use bioreactors. Now, there's very obvious advantages of using plates instead of a bioreactor system, but there's also some disadvantages. So that would be, there's a limited control over your culture environment in plates, and this may lead to questionable relevance of the fermentations in plates compared to an industrial setting. It's also challenging to achieve a metabolic steady state in plates, which is what's quite important for metabolic modeling. And finally, there's limited material available in the plates for sampling. So to calculate fluxes, which is needed for modeling, we need repeated omics measurements. And we also need additional sample for metabolomics and proteomics. So bioreactors obviously have plenty of sample available. They're highly reproducible. You can have strict control over the culture environment and feeding strategies, and therefore they can more closely mimic the industrial conditions. But there's very obvious disadvantages, including the high cost and mostly the throughput, right? So your throughput will be limited by the longer culture conditions and the number of bioreactors that you have. So for our Amherst collaboration, we purchased 16 bioreactors from Inforce, so 8 could be run in parallel at a time. We had online optical density measurement, temperature control, pH stirring, dissolved oxygen, and gas control and measurement. We also had full integration with a hardened GCMS for online off-gas analysis. And this particular reactor system came with this Eve software, which allowed readily integration with a LIMS system. And that allowed us to test 16 designs a week. So for analytics, we combined with Queensland Metabolomics and Proteomics, which is a group that was funded by the federal government to allow ready access to scientists and industry to these sorts of analytics. So they had a lot of expertise and cutting edge instruments, but what it didn't have was really the capacity to do all the sample processing, so pre-processing of the samples. So we invested in automation here. We used a Hamilton Starlet robot to automate proteomic sample. So that was the tryptic digestion using the S-trap sort of protocol. And we also got a Flowbot and some Integra robots just for simple pipetting, serial dilutions, solvent-solvent extraction, format changing, that sort of thing. We then used an open source LIMS system called OpenBIS. So that allowed us to integrate all our bioreactor data that was taken out of Eve, as well as all the data from all our different analytical instruments, be it HPLC, LCMS, that sort of thing. So here's an example of just all the metadata from the bioreactor samples. We have everything from OD, time of measurement, which reactor it was, the volume of the sample, everything you'll need to know. And this, we can very readily query into OpenBIS and extract all the information required. So this, for example, is all the HPLC data associated with a particular sample. So just a brief overview of our software workflow. We extract all the data from our bioreactors, all our omics data. We pool all of this in our LIMS system, OpenBIS. We can query into it and extract all the information in a CSV file or an Excel output. We then read this into Python, did our pre-processing of the data and got it in a format required for our metabolic modeling. And then we did the modeling itself in MATLAB. So now I guess the focus of the talk is how we learn from this omics data. So to be able to improve our designs, we really need to understand what the rate limiting step is in a particular pathway. This itself requires knowledge of the kinetics. But on the flip side, enzyme kinetics are typically unknown, and what we can access through, for example, Brenda, is not necessarily reflective of what's actually occurring in vivo. So what we really need is kinetic models, which allow us to integrate metabolomics and proteomics data. And we can use this then to sample and estimate what these kinetic parameters are. This will give us a whole set of models which could potentially explain the behavior that we see for a particular strain or a big population of models. And we can do simulations with this population of models, usually metabolic control analysis, and use that to predict what the rate limiting steps are. This is a sort of brief summary of the software workflow here. And we can further refine the set of models by then testing it against other conditions or other strains. So just how exactly is the omics data used? Well, the flux or the rate of a reaction is a function of the driving force of the reaction and the capacity for the reaction. The driving force of a reaction is a thermodynamic driving force, and the capacity, of course, is the amount of protein that's present. And these two things are dependent on omics data. So your thermodynamics is a function of your intracellular metabolite concentrations. And of course, enzyme capacity can be measured through proteomics or approximated with transcriptomics. Your flux itself can be inferred through metabolic modeling. We can combine all these things together to generate a population of kinetic models, and we can use these kinetic models to then predict how to modify our system to improve production of our product of interest. So how do we actually step through this? Well, you probably start with a single strain, or you can start at least with a single strain. You infer your fluxes, you measure your metabolomics data, and you can sample thousands to tens of thousands of models and use metabolic control analysis to predict your rate limiting steps. From this, you can create a set of designs to address these rate limiting steps, and you can test them with the biofoundry. You can build your strains. And then again, you can characterize these strains. This time you need to infer your fluxes and measure metabolomics and proteomics data. And with this, you can then start to prune your population of models down to those that describe all of these new strains. With this, you can then use metabolic control analysis again, or simulation of these kinetic models, because at this stage, there's a much smaller population of models. And the cycle continues. So the software we use for this is called GRASP. I'm not going to bother with the details of it. It's very similar to what we presented on the other side. So just to illustrate this through our case study, working with Emrys. So they gave us 2 compounds out of their 450 molecules that they were given under the DARPA Living Foundries 1,000 Molecules Initiative. So the discussion about the molecule itself is going to be anonymized for the sake of Emrys. But to describe the collaboration, so Emrys supplied us the base strains, and what we asked for were actually challenging products. So products that their current biofoundry workflow weren't able to progress any further. And they advised us on how to set up the fermentations, and they built our strain suggestions. So for this particular product, they built 88 strains for us over 2 separate design rounds. And then they performed a scale-up test for us in their bioreactor of the best performing strain. So we then were responsible for testing the strains in the bioreactors, performing phenotypic characterization, and performing model-guided rational design. So to show you the kinetic modeling in action, I guess, let me step back and briefly describe this particular problem for their compound. Emrys had actually already done an amazing job. They had a reference strain that produced a product of interest at the grams per liter titer. The problem was that the precursor for that product, which requires a methyl transferase to act on it to make the product of interest, was 3 times more abundant in the culture media. So there was a limitation in this methyl transferase step. So the major focus for us was how to actually increase the methylation of this precursor compound. Now, the design scope of this was enormous because it's associated with 1-carbon metabolism. So we're studying over 50 relevant genes, including the pathway of interest, the S-adenosyl methionine cycle, which is responsible for the methyl transferase activity, and the 1-carbon metabolism itself, which sources the tetrahydrofolate groups for the SAM cycle. So our initial designs could take up to 12 gene insertions, and we can do 2 separate design rounds. So this is the results of our metabolic control analysis. So for our precursor product, we could easily see there's some enzymes in the pathway which are limiting the production of the precursor, and we actually tested overexpression of these, and we got some positive results, but that wasn't really the goal. The goal was how to increase the product. Now, non-surprisingly, there's a lot of control associated with the methyl transferase step, but there's also a few hits in the SAM cycle itself. So our initial designs focused on overexpressing the SAM cycle, and we also increased the rational design space a little bit to target 1-carbon metabolism. So out of the 24 strains that were built, verified, and for this out of this first design round, 14 showed improved performance in terms of having a higher titer. Some worked quite well, having great yields and reduced ratio of the precursor to product, but they didn't give a high titer, so that's not really a success, right? So just to show some of the omics characterization of these strains, we have metabolomics data on the left, proteomics on the right. The columns represent metabolites and proteins, and the rows are strains. So what we can see is our genetic changes were changing metabolism. We're getting quite different concentrations of metabolites and different abundances of the proteins, which not only shows that something's changing, but it gives us great data for the modeling. So we can then really refine the models with this sort of data. So we then actually expanded the kinetic model out of round one to capture 1 carbon metabolism. And we actually saw a lot of the control was now sitting in 1 carbon metabolism. So we really should have considered this from the get-go. So subsequent design rounds, round 2 really focused on trying to increase the expression of 1 carbon metabolism. Our best strain out of here, 366. So it was based off one of the high performing strains out of round 1. And it was the actual design was based on our GRASP predictions for 1 carbon metabolism. So we tested our top performing strains at AIBM. We found the best performing strain, and Amyris tested it in a 2-liter scale-up. And it reached 11 grams per liter titer, which was about a 3-fold increase over what they were getting with their reference strain. So that was a nice success. What we could see is even after trying to overexpress the SAM cycle and 1 carbon metabolism, 1 carbon metabolism still was limiting. And a lot of the strategies that we tested were quite different and showed modest improvements. So future design rounds could try and incorporate these different designs together. So we have created a service called IDEA Bio, which is the integrated design environment for advanced biomanufacturing. So we're offering the sort of collaboration that we've had with Amyris now as a general service. Just to give you an idea of the sort of scale of projects we're tackling, back in 2010, we characterized 4 strains for Dow. We've done 250 strains for Amyris in 2020, and we're about to try and characterize a genetic knockout library for LanzaTech, consisting of 2,500 strains. So I'd like to thank the following people, particularly AXA for doing all the fermentations, and the Amyris team, which is not limited, but includes Sunil, Kirsten Benjamin, John Hung, Adam Meadows, and all the funding bodies on the right here. And here's my email if you have any questions about our workflow or you want to collaborate or whatever. Thanks for listening.
I think that the next presentation is online.
Hello, can you hear us? Are we sounding fine?
Yes. Yes.
Okay.
So, so the next presentation is going to be, is coming from Sky Biofoundry in South Korea. And they're going to be talking about deep learning with applications to DNA assembly.
Hello, I'm Sung Chong Ko.
And I'm Dong-Jae Kim.
And we are graduate student in Dr. Ru's lab at Sungkyunkwan University, South Korea. And today we'll be presenting Biofoundry Pilot and its application for the Biofoundry workflow, DNA assembly, and deep learning framework. Before we start out, we want to give us a shout out to our PI, Dr. Han Min-woo, who is the director of Sky Biofoundry. And we also want to thank the Global Biofoundry Alliance for giving us the opportunity to speak at this wonderful conference. Our school and Biofoundry is located in Suwon, Korea. Suwon is located just 20 kilometers away from Seoul. And this geographical adjacency give us the advantage for being able to cooperate with major chemical and biotechnical corporation in Korea. The establishment of our Biofoundry was assisted by government funding. And shortly after, we joined the Alliance for Cooperation and Possible Contributions. We are not an industrial scale foundry. So our foundry definitely has its limitation in its scale capacities. However, we are consistently working towards optimizing our workflow within our system to streamline our research process. And our meaningful results pipelines. These are the present and future pipeline we have established for our system. The first is the genome engineering pipeline, which we utilize for strain genome engineering. We made steady progress on optimizing CRISPR-Cas-based genome editing system for automation workflow. The second is the DNA assembly pipeline, for which we modularized and organized modular cloning systems for synthetic biology approaches. Lastly, we are also currently working on developing protein engineering pipelines using our automation platform so that multiple mutants can be generated, assessed, and applied in a high throughput manner. We find that every biofoundry should be equipped and prepared with tools that allow the smooth progression of the DBTL cycle. Annotated here are just a couple of the instruments and tools we have. Data generated in well-placed scales can that be used to learn patterns and models. And we've had a case where we used automation to validate our models data. We organize our lab automation procedures as follows. Workflow refers to the overall goal of a single automation process, whether it may be cloning, pathway construction, genome engineering, etc. To achieve the goals of each workflow, we then further break it down into progress modules or step, which you can interpret as a single lab procedure. This includes PCR reaction, enzymatic reaction, transformation, and colony selection, colony picking, and such. For the execution of these steps, relevant robotic units are required. For example, to conduct a PCR reaction in an automated fashion, an on-deck thermocycler is required for the reaction, and a vacuum unit is required for purification of the product. To go even further, we've added an additional hierarchy to the schematic and labeled it as a before experiment unit, or BU for short. For short, BU refers to the consumable components and the components that directly deal with the disposable. For example, liquid handle tips and tip legs that contain these liquid handle tips are categorized under BU. Not only that, the positions at which the tip legs should be placed on the platform is relevant for BU consideration. We want to explain why placing consideration into managing at the BU level is of importance through an example. Let's take the step 3 molecular cloning as our example. Cloning itself is broken down into steps. Cloning consists of 5 steps. 1, PCR reaction and enzyme restriction, E. coli transformation, colony selection and inoculation, and plasmid purification. Step 3, which corresponds to E. coli transformation, can be further broken down into 2 process modules. Ligation reaction of DNA fragments and E. coli transformation. The robotic unit required is the EU platform or heater or striker. As of the consumable units, which is our BEU, the identities correspond to enzyme restriction products, E. coli competent cell, 5x KCM transformation buffer, Petri dishes. And when taking into account the quantities available of the BEU, we have the following. For a set number of samples, for each identity, we have the corresponding volumes. For each corresponding volumes, the vessel and consequently the position of the vessel vary accordingly as well. Depending on the identity and the volume, they can be contained in tube, trough, strikers. And the location of the EVO platform has to address will vary depending on the format. And now I'm going to hand over the floor to my colleague Dongjae, where he will explain the definition of BU, palette, and its applications.
Hi, so I'm Dongjae, and I'll be continuing with the presentation. So, in summary, BEU not only encompasses the quantities of the consumables, but also their identities as well as their position of localization on the platform during operation of the automated procedure. So when BEU represented as a function also depends on the number of samples, which may vary even while operating for the same workflow. Palette takes all these parameters into consideration and organizes them in a user-friendly manner. So why did we think a tool such as Palette was needed? Like, why not just use Excel or manually calculate the BU every time? It's because when actually operating multi-stepped automation workflows, BU compositions can grow increasingly complex, and the use of a computer-aided organization program greatly reduces margins for human errors. So as an example of how complex BUs can be during actual experimentation, here we present two cloning cases. The case on the left is the construction of 24 different unique CRISPR-Cas guide RNA plasmids. As presented here, the number of troughs, tubes, tips, plates, and stackers vary per step. And the required quantities will differ if the number of samples will vary from 24. So the same goes for the case on the right, which corresponds to pathway constructions, and the same complexity exists for this case as well. So BioFoundry Palette allows the streamlined determination of DEU parameters and outputs to the user a simplified table of the required parameters per step. This greatly simplifies the preparation process, or DEU, of automated lab experiments, which greatly lowers the entry barrier for employees and students that are new to automation systems. The program was built on Python and outputs as an Excel file, which contains information on the DEUs required for a given step. We've packaged the program into an executable file and can be downloaded through GitHub in our link over here. So as a proof of concept, we want to introduce a couple cases where Palette came with great assistance to us. In one of our recent works in early this year, we conformed MoClo, or Golden Gate assembly-based modular cloning, to our robotic platform, and just calling it RoboMoClo. As illustrated in our paper published in 2022, we utilized the platform to generate pathway units, or 8-gene operon scale expression vectors, as illustrated over here, using almost entirely automated procedures from basic units such as primers and single gene harboring plasmids. So we modularized the operational procedure as well as the expressional units, ranging from single gene TUs or transcriptional units to multi-gene subpathway units and pathway units. So as illustrated in this diagram, using RoboMoClo to build up pathway unit constructs required the operation of various steps and multiple repetitions of workflows with sample size varying per iteration. So as a consequence, BU parameters were subject to constant variability multiple times. Using Palette, we were able to smoothly carry out the entire process with minimal human-induced errors. So our next example is a work that we concluded early this year as well. We constructed a model based on open source data and built a model with deep learning. The model was set to predict the translational efficiency of proteins incurred by changes in translational elongation short ramp sequences, which consists of the 3rd to the 5th codons of a coding sequence of an mRNA. So building the model was one thing, but biologically validating the model was another. Various test-R sequences and their expression scores had to be validated through cloning, right? So our automation platform was used for this purpose. Palette was utilized for this experiment, greatly enhancing the construction process of the plasmids required for this purpose. And lastly, though yet unpublished, we've also applied Palette into guide RNA cloning. and consequent CRISPR-based genome editing, such as base editing and deletion mutants. We've successfully created a library of around 2,000 guide RNAs, and BU determination by Palette was again a key success factor to its efficient construction. So we want to summarize by saying that using Palette assisted us greatly in smoothing automation experiments in our lab. We believe that using Palette or another program more catered to your lab, but still focused on managing BUs, should also deliver the same effect. We want to remind you that a lot of the times, especially for small biofoundries like ours, 100% automation is really difficult to attain due to practical issues. At least for us, that was really the case. Many of the procedures required to execute the automation itself is often carried out manually by human operators, and Palette allowed us to reduce errors, ameliorate tediousness, and increase time efficiency for these manual processes. So to end things up, we want to thank collaborators, mentors, and SKKU FMB lab members, and also to our director, Dr. Hanmin Wu, for allowing this work to be developed. And we want to thank our sponsors, such as Korean taxpayers, for funding our research. And thank you very much for listening, and we hope that our approach can provide valuable insights for your research as well. Thank you.
Thank you very much. The next presentation is online as well. It's coming from Singapore, and Dr. Yo is going to propose a unified modeling approach to the design cycle for biofoundries. Can you hear us?
Yes, I can hear you. Yeah, okay, I would like to share my screen. Can you see my slide? Yeah. Okay. Yeah. Good day, everyone. I'm actually I'm using Wei from NUS Engineering Biology Lab, and we are actually affiliated with the NUS City. And today I would like to talk about a unified modeling tool to enable the model driven engineering design cycle for biofoundry. So without further ado, so as you all know, in synthetic biology, we have to go through the standard design, build, test, learn cycle in an iterative manner in order to achieve the desired performance for our genetic circuit design and optimization. So to expedite the process, modeling has played an important role in particularly for the design and the learn phases. In the way of it can be in the form of reverse engineering, where we can actually extract the knowledge from the experimental observation for hypothesis testing, and also we can also like perform the forward engineering to make use the model to make prediction, and also to apply the model driven approach to guide the experimental design. However, there's always challenging for the model identification, especially when we want to derive an appropriate model with good explanatory or predictive power. So in this case, we always have to repeatedly go through the modeling cycle, starting from like identifying the modeling purpose, deriving the model structure, estimating the parameter, followed by model analysis and optimization to optimize the model, and repeat the cycle again. So it is important for us to expedite the process and to prevent the real reinvention. So besides, there's also a need for us to repeatedly performing the hypothesis testing in order to identify the good model that can actually well capture the different expression profile. However, interestingly, we find that most of the expression profile are usually conserved across the similar regulatory system. It is therefore crucial for us to have a model bank that can be used to store the model for future hypothesis testing. So in general, for the efficient model discrimination is always required the parsimonious model, which are the simple model that can actually explain the data using a minimum parameters. And we also require the optimal experimental design that can actually provide the maximum information gain. It matters because the ear-defined parameter could actually significantly affect the model predictability capability and also affect the quality of the insights that we can actually derive from the model. So this can actually be addressed by implementing the identifiability analysis known as IA in order to determine if the variable or the parameter are actually identifiable based on the measure variable or data, experimental data. So in general, there are two types of IA analysis. One is the structural identifiability, and the other is the practical identifiability. So for the structural identifiability, is mainly arises from the model structure and based on the specified measure, the specified variable to be measured, and it can actually provide insights into the interdependency between the variable and the parameter, and it is mainly independent of the data quality. So we can actually run this analysis even before we run the actual experiment. And the insights that we get from these structural identifiability analysis can then be used to guide the model reformulation or for us to plan the variables to be measured experimentally. On the other hand, for the practical identifiability, is caused by the noisy or insufficient data or even the low quality data, and it can actually help to examine the confidence interval of the parameter estimate, and is usually run after the actual experiment. And then this can be actually improved by adding enough high quality data. So a good model that can actually provide the good explanatory power should be both structurally and practically identifiable. Despite the usefulness of this analysis tool in the model selection process, however, it is currently still lacking in most of the modeling tool. So here we have develop a unified modeling tool known as a biomodel selection system, BMSS for short. And this is actually a revised version that aims to streamline the model selection process with the necessary with the essential the numerical methods, the analysis tool, and also the information criteria that is actually required for model selection process. And more importantly, this tool is also database driven, where we can actually support the model storage and retrieval for multiple model comparison, and then to be used in the model selection process. So essentially, this tool allows us to take in the model input in the form of a simple configuration file or in the form of the standard SBML file supported by other external software as well, or database. And then we also have a built-in databases known as MBase or UBase. And one is actually the global copy, and the other is actually the local copy. So after importing the model and also the setting specification, and then curve fitting can be actually implemented on multiple model for parameter estimation based on the imported experimental data. And then the model ranking can, based on the information criteria, can then be used to compare the performance of the different model based on their goodness of fit or the model complexity. And then aside from that, we can also implement the structural identifiability analysis to guide the model reformulation or to plan for the variables to be measured experimentally. And then we can also implement the a posteriori identifiability analysis based on the trace analysis to verify the conversion of the different parameters. So sensitivity analysis can then be performed on the best chosen model in order to identify the sensitive parameter that can be used for tuning, or to identify the correlation or interdependency of the different parameters. And then the user can then proceed to run the simulation or make prediction in order to guide the experimental design. So to demonstrate the utilities of these two, and we actually, here is a simple case study. So we actually make use of this simple logic gate system from earlier literature. And then we are, in this case, we are expressing the fluorescence protein driven by two inducible promoter. And then one promoter PTAT is actually induced in the presence of ATC, and the other PBAT is actually induced in the presence of arabinose. And here is the characterization data obtained from the microplate reader to capture the expression profile at 4 different logic state. And this line represent the result for the logic state in the absence of both inducer, and the 0, 1 represent the state for in the presence of ATC, which actually show an increasing expression profile followed by a slightly decreasing trend. And the 1, 0 is actually the case for in the presence of arabinose, which actually exhibit an exponentially increasing expression profile. And lastly, the 1, 1, 1 logic state is actually represented by this rate expression profile. So in this first iteration, so we have developed 11 model, which can where each of them actually carry different underlying hypotheses. And then from the model selection and the ranking process, and model 1, which incorporate the hypothesis of like inducer degradation to capture the profile of 0, 1 logic state, and the delay induction to capture the 1, 0 logic state, and also the resource competition hypothesis to capture the profile of 1, 1 logic state, and has been ranked as the best model by the AIC, the Akake information criteria. So from this AIC ranking table, we can actually observe that the other 10 model, they actually indicated no support for the evidence, which also means that their performance are actually not comparable to the top ranked model, because the AIC difference is actually more than 10. And what we can observe here is the performance for this model, model 1, they actually well capture the expression profile. So we then proceed to perform the a priori structural identifiability analysis on this best chosen, the top ranked model 1. And then this model actually consists of 9 variables and 10 parameters. And in this case, we assume that all the protein variable are to be measured experimentally. And however, surprisingly, despite the good fit that we observed in the earlier slide, and in this case, 2 out of 9 of the state variable and 7 out of 10 of the parameters were found to be non-identifiable based on these measure state variable. And then we move on to assume that what if all the state are to be measured in this case. So, but even so, we also from this analysis we also identify that 40% of the parameters remain non-identifiable structurally. And then to further analyze the parameter identifiability, we then perform the a posteriori IA analysis based on the trace analysis. So this is done by running multiple times of the curve fitting, and then analyze their trace analysis for the parameter conversion. And then in this case, for these 3 different runs, we can actually see that they actually all of them actually well capture the experimental profile. However, from the trace analysis, most of them, most of the parameter are actually not well converged to a single unique solution. So aside from that, and we can also perform the scientific analysis to help us to provide more insights in this case. So aside from providing the aside from identifying the sensitive parameter for the individual individual parameter against the in response to the output, and then the second order scientific analysis can actually be used to identify the correlation among the different parameters. So from this analysis result, we actually observed that most of the parameters are actually highly correlated. And this also suggests that there could be some redundancy in the number of parameters used in this model. So in the second iteration, then we then proceed to reduce the model guided by using the a priori structural IA analysis. And then in this case, we have by using this method, we have actually come up with 4 simplified model, and together with the top rank model from the first iteration as the control model for comparison. And then from this model selection, and then we identified that the simplified model 4 with only 6 state variable and also the 6 parameter has been ranked the top by using the AIC ranking table, and the other 4 model indicated no support for the evidence, mean that they are actually their performance is actually not comparable as the top rank model. Yes, so in this case, yeah, we can see that the is actually well captured as parameter profile. And then we move on to perform the structural IA again for this simplified model 4 by assuming that all the protein state variable are to be measured, and then we still found that 1 state variable and 1 parameter are still structurally non-identifiable. So based on some analysis, then we assume that two additional data regarding the inducer concentration are to be measured in this case, and then with this assumption, and then we observe that all the state variable and also the parameter are now becoming structurally identifiable. So this is actually the trace analysis result before adding the 2 additional data for the inducer, and then 4 parameters still fail to converge, as can be observed visually using this trace analysis, or it can also be computed quantitatively by using the large R hat value. So R hat value is actually to calculate the variance for the from the different traces. And then as a side note, we are also keen to know how the identifiability analysis can actually affect the model predictive capability. So in this case, then we try to make use of a fully identified model to generate a synthetic data for testing purpose. So in this case, after fitting the model to the original data, and then we use the model to simulate the concentration profile for the 2 inducer, and also use the model to predict the condition when the protein degradation rate increased by 1.5 times. So in this case, we observed that the simulated profile are actually quite different from the synthetic data, and there are also some deviation in terms of their model prediction. Then we move on to verify our insights by fitting the model aside from to the original data. We also fit the model together with the 2 additional synthetic data for the 2 inducer. And then in this case, all the parameters finally managed to achieve a full conversion to a single unit solution. And then more importantly, they actually show their model prediction actually show a very good agreement with the synthetic data when changing the protein degradation rate. So from here we have demonstrated how the different analysis could be actually used to drive the model selection process, and how the identifiability analysis can be actually used to improve the model predictive power. So this is actually an open source Python tool is currently available for downloading from GitHub. And then to ease the use of these tool, and then we also are developing the GUI to support the model selection process. And then in this case, we actually support the import of the multiple experimental data, and then the user can also choose multiple model from the database or upload their new model in the form of configuration or SBML file, and then the user will need to link the different data to the different state variable from the model, and then the GUI will actually output the model ranking table and also the best best rank model together with the corresponding parameter value. And then definitely the user will be able to visualize the model fitting performance for the different model. So we also have a web page for the Embase, where the user can actually browse the model and also download the model configuration file in order to be implemented in the this BMSS2 platform. And it can also download the model in the form of SBML format. And then for now, we have a different model which actually capture the different underlying hypotheses for the regulatory system, including the inducible, constitutive, and the different logic gates. And we also have the different growth model and also the resource aware model. So we envision that this library would be extended with more usage of these two. And then from our side, definitely we are going to extend this model list. And then the next question will be how these model-driven modeling tool can actually be useful for the biofoundry application. So in this case, we can actually make use of these modeling tool to perform the in silico screening for combinatorial study. So we can actually have a model that incorporate the metabolic network and also the resource-aware model with different proteome pool. And then we can also have the model for the genetic circuit to describe the different enzyme expression and also a bioconversion pathway. And then with a list of or a library of a promoter or RBS with different strength, and then a large combination of the construct can then be screened in silico. And then this construct can then be ranked for the performance based on the output yield or even their chances for failure in the construction phase. And then a few of these constructs can then be with different performance level can then be selected for to proceed with the construction building. And then the data that we obtain from here can then be used for model validation. And then we can repeat this cycle for a few iterations until we achieve the construct with the desired performance. So in short, this model driven approach will allow one to actually narrow the design space and also to provide the quantitative precision. And then aside from that, the combination that generated from the screening process can then be pumped into this high throughput plasmid assembly designer. This is another in-house tools known as HiPad. And then this tool can actually support the primer design and optimization, and can also provide the assembly route optimization. And then with this GUI, it can actually take in the task file that contain with the different combination or with the different part list, and together with their the input sequence. And then it will actually output a list of the different primer design required for the assembly process, and also the different plasmid variants, and together with the optimized assembly protocol. And then this can then be linked to some automation instrument for the hardware setup. So yeah, just to sum up, just to summarize. So what is the role of these two in biofoundry? So I have presented these two, a unified modeling tool known as the BMSS tool. And then this tool is actually aimed to streamline the model selection and analysis process. And then in this tool, we also make use of one of the GBA tool from the SynBioPython package for standard file generation, such as the SBML or the combined archive. And just a quick note. This SynBioPython is an open source package developed by a few of us from the GBA software team in an attempt to create a standard tools that can be shared by the community. And then the combination that generated from this tool can then be passed into this HiPad assembly tool for plasmid design, and to generate the optimized assembly protocol to be then linked to the automation instrument available in the biofoundry. And then in this case, we can also make use of another the lab automation tool from the SynBioPython contributed by Edinburgh Genome Foundry in order to generate the pick list and the protocol for to automate the some of the instrument for the experiment. And then the data that we actually get from this biofoundry can then be fed back into the BMSS2, and then the output from these and then can be used to as a model-driven approach to optimize the experimental design. So yeah, that's all for my presentation today. So here I would like to share some of the useful links. So here is the GitHub repository for the BMSS2 and also SynBioPython. And these are the publication for these BMSS2 and also SynBioPython package. And this is the link for the Embase web page. And then if you are interested to get to know more the research done in our lab, you can actually refer to this engineering biology lab web page. And then if you have any question, you can actually contact me or our lab PI, Prof. Potrelu. And then I would like to thank some of the contributor for developing the package and also GUI. So these are the contributor for the BMSS2. And also I would like to thank our PI, the Prof. And also some of the contributor for the SynBioPython package. So yeah, that's all for today. Yeah, thank you. Any question, let me know. Yeah.
So thank you very much for this very exciting talk. Thank you very much to all the speakers, because we managed to finish on time, which is remarkable. I think that Vincent is going to tell us more about what the rest